Welcome to Concept Map Summary, and our topic for this time will be carbohydrates. Carbohydrates. We are required to have at least 60% of carbohydrates in our diet. That is why 50% of the diet of most people in the world constitutes carbohydrates. Carbohydrates are found in different forms. For example, in South America, their carbohydrates or staple food comes from corn. That is also found in potato and wheat in the people of the North America, while this is equivalent to cassava in some parts of Africa. And generally here in Asia, the staple food or the source of carbohydrates is coming from rice. Carbohydrates. An example to this is natural sugar. Natural sugar are found in milk and fruits. Another example of carbohydrates is refined sugar. These are actually sugars that are extracted from a plant source, specifically sugar canes. So what are the different types of carbohydrates? The first type of carbohydrates or sugar is called monosaccharide. In other words, it has only one sugar base unit. An example to this is deoxyribose. A deoxyribose is actually a component of a DNA, or known as deoxyribonucleic acid. When we speak of deoxy, meaning this sugar has no oxygen that is attached to it. Another type of sugar is called a ribose. When we speak of ribose, it has an oxygen attached to it. A ribose is part of an RNA, or ribonucleic acid. Now, monosaccharides are basically energy sources. When we speak of energy sources, they are readily available energy, such as glucose and a galactose. Now let us move on to the other type of sugar, which is this time two sugar base units. In other words, it is basically called a disaccharide. The examples are lactose. Lactose is basically made of two sugar base units. These are glucose and galactose. The combination of or the linking between glucose and galactose is called a glycosidic bond. An example of lactose is the sugar that is found in milk. Another type of disaccharide is maltose. This is glycosidically bonded between glucose and another glucose. Maltose are sugars that can be found in malt. Malt is used in beer making. The third in my example for a disaccharide is sucrose. Sucrose is a formation between glucose and fructose. Sucrose is a sugar that are found in plants. Now let's move on to the next classification of sugar, which is called oligosaccharides. Oligosaccharides are made basically between 3 to 10 sugar base units. That is 3 or more sugar units. An example to this is glycolipid. These are sugar units that are attached to our lipids. Another one is glycoproteins. These are sugar that are attached to our proteins. Then the last one here is called polysaccharides. This ranges from 100 to 1000 of sugar base units. Polysaccharides are now used in structures. An example to this is chitin. Chitin is what makes the exoskeleton of crabs and beetles. In plants, a cellulose is an example of a polysaccharide. Another function of a polysaccharide is basically to store energy. In animals, energy can be stored in the form of glycogen. Like Glycogen can be found in our skeletal muscle and our liver. You know that plants basically is the primary producer of energy through photosynthesis. The energy that is stored in plants is through starch. Example, starches that can be found in potato and rice. When two carbohydrate units will be attached, they are bonded together through what we call a glycosidic bond. This is one sugar unit and this is another sugar unit, and when we attach the two sugar units together, we will extract H2O and making oxygen as the glycosidic bond. This is what a glycosidic bond is. Again, glycosidic bond is linked by what we call an oxygen atom. During the glycosidic bond, H2O is being released. Glycosidic bond occurs in the OH group in one of the monosaccharides. This OH group in the monosaccharide is called the hemiacetal group. Here are some examples of a glycosidic bond. First is maltose. It is glycosidically bonded between glucose and another glucose. It has a structure of alpha-D-glucose and a D-glucose. To break maltose to glucose and another glucose, this will require an enzyme called maltase. Maltase will break down this glycosidic bond into two glucose molecules. Maltose, by the way, is produced when we break down starch. Starch is a type of a polysaccharide. In humans, we can break down starch through digestion. In plants, we can break down starch through seed germination. Another glycosidic bond is called lactose. This is glycosidically bonded between galactose and a glucose. Lactose is made by the beta-D-glucose and a D-glucose. The lactose glycosidic bond can be broken down by an enzyme called a lactase. Lactose is produced in a mammary gland. The last of my example is sucrose. It is glycosidically bonded between a glucose and a fructose. Sucrose is made from alpha-D-glucose and beta-D-fructose. The glycosidic bond between fructose can be broken down by an enzyme called sucrase. Let's go back to oligosaccharides. Here are some examples of an oligosaccharide. Tetrasaccharide. An example of a tetrasaccharide is a stachyose. A stachyose is an artificial sweetener. It contains the component of alpha-D-galactose, beta-D-fructose, and alpha-D-glucose. Another one is trisaccharide. An example is raffinose. This is very important to plants. It contains fructose, a glucose, and a galactose. An example function of this oligosaccharide is called an antigen. This antigen can be attached to our red blood cell. These are the different red blood cell types. So for type O, this is the oligosaccharide that is attached to O that will determine O as an antigen of O or blood type O. For blood type B, this is the oligosaccharide or oligosaccharides that are attached to B. And this is the antigen that is attached to type A, red blood cell. And this is the antigen or the oligosaccharide that is attached to blood type AB. This antigen is responsible for determining our blood type. Polysaccharides, also known as glycan. Our polysaccharides or the glycans can be identified through the following characteristics. The first one is our monosaccharides are always in a repeating unit. There are two types of a repeating unit of our polysaccharide. One can be a type called heteropolysaccharides, meaning this contains two or more types of polysaccharides. An example to this is hyaluronic acid and heparamine. This is an example of a repeating unit called heparamine. Another type of monosaccharide repeating unit is called homopolysaccharide. When we speak of homo, contrary to hetero, homo means only one sugar repeating units. An example to these are glycogen, starch, cellulose, and chitin. This is the molecular structure of a chitin. It is basically homo because it has the same type of sugar units that are being repeated. And chitin basically are the exoskeletons of our different crustaceans and insects. The second identification characteristic of a polysaccharide is that the polysaccharide usually has a length of polymer chain from hundreds to even millions of chain units. The third identification characteristic of a polysaccharide is the type of glycosidic linkage between the monomers of our sugar units. And finally, the type of degree of branching of our polymer chain.